My name is Daniel Chiu. I'm at the University of Washington in Seattle, Department of Chemistry and Bioengineering. So we predominantly develop technologies, and then we're trying to use these technologies to analyze semen exosomes in detail. So the technology that we develop are threefold. One is a single molecule flow method. Another one is a fast sorter to sort the EV's exosomes. And then the third one is to use an imaging digital PCR platform we developed to try to look at the nucleic acid contents of the vesicles. It fits it into two ways. One is that as a group we develop a number of different technologies. So single molecule methods, microfluidics, optical instrumentations, new types of fluorescent probes. So this EV project nicely tied together many of the technology areas that we work in. Second, it also ties in because on the more biological biophysics end, we've been very interested to study individual synaptic vesicles. It is something that we've been doing since 2000, since I started my lab for over 10 years. And so a synaptic vesicle is 40 nanometer to 50 nanometer thereabouts. They're pretty homogeneous in size. They are very abundant in the brain, so you can do nice preps for them. And so we study them quite extensively. And so, from the size scale and the number of molecule perspective, it's actually very much like EV, although we're very much new to the EV, per se community. So how I got interested in synaptic vesicles. So I actually, I've always interested in neuroscience. My undergraduate degree was in neuroscience first, before I did another degree in chemistry and a more physical chemistry. And so through that, it's always sort of maintained a strong interest in neuroscience. And then even in my PhD, I was sort of doing a project together with a chemistry group at Stanford and the neuroscience group, Richard Scheller, trying to study vesicle contents and stuff like that, and trying to develop new methods to study them. So that's a very long-term interest that I have, and we continue to do that and try to study something in great detail. In terms of how that ties into EV, I guess we didn't sort of go in wanting to study the EV. It's just that we read more and more about it. They are very heterogeneous. It seems like some of the things that we develop could be used towards it. And so we're kind of drawn to it because of the tools that we have, and not per se that we go in wanting to know everything about EV. But we hope that the tools that we develop will be enabling or complements the tools that people have, so that we can understand it better. Synaptic vesicles, there's a finite number of vesicle proteins. They're easily grouped together they're not easily grouped together, but you have different vesicles, oftentimes classified based on the transmitters that they have. So they're all very monodispersed in size. So they're not heterogeneous in comparison. Everything is relative. So in comparison to EV, they're not heterogeneous given the broad distributions in size, cell of origin, tissue of origin, different combinations of markers. So I think EVs are very, very heterogeneous, and there's lots and lots of subpopulation, which is why we're excited to sort of develop a single molecule method to really subtype all the EVs there are in a high throughput way. So this is, we'll see how far we can get, but this is at least is a motivation for us. For the single molecule flow device and sorting device, we bring together a couple different technological areas that we have developed. One is optical instrumentation. So we've been working in the past on a single molecule continuous flow capillary electrophoresis to try to separate individual molecule by molecule. And so we have two laser beams. We do cross correlation to pull out the retention times, and through that we developed a line confocal method that allow us to have good throughput and while maintaining good single molecule sensitivity. We developed some new fluorescent probes that are brighter, so that also helps. And also we developed microfluidic channel systems such that we can spatially confine the EVs to match the laser probe volume to get maximum sensitivity, and also design different channel structures and actuation mechanisms so that we can sort individual EVs very quickly, because unlike sorting cells, sorting EVs, you need to sort a lot more because the mass is a lot less if you want to do downstream mass spectrometry or omics or sequencing and so on. The idea there is that if you look back in the single molecule detection, one of the way that was done, and this is something I did for my PhD, is to use a confocal, very tightly focused diffraction limited volume to detect individual molecules. So it is a little bit of a contradiction there, because you get maximal sensitivity when your laser volume is the smallest, because that lowers the background. Because for a molecule, there's only so many photons that you can squeeze out. And so because of that, and because the volume is very tiny that you focus down, you need to have high concentrations of molecules, because otherwise the sampling time from one molecule to another is just very long. And so how do you try to sample more molecules, or in the case of a microfluidics, be able to have larger cross-section channels and still not miss any channels, so miss any molecules. So the need is that you need to cover larger cross-sections as the molecules flow through. And so a compromise that we found is to basically have a line of light that cuts across the whole channel, that minimizes background because there's not a round big spot, but it's a very, very thin line. But then also you don't miss any molecules as they go through the channel. So that's the compromise we found in terms of what is the best optical geometry, what is the best microfluidic geometry that could match with that, and so on. Digital PCR has been very popular. But the most successful probably is a droplet digital PCR. So we developed a platform where we have a static array of digitized volumes, and the advantage there is that unlike a flow-through method, for many of the droplet digital PCR for detection, we do imaging, and it's a flat surface, so we can do imaging quite well. And so the advantage that provided us is that we can image the sample before doing digital PCR, we can image during digital PCR, and we can image after digital PCR. And so specifically for EVs, that is advantageous, because then we can correlate how many EVs that we have and what kind as we go through the process of digital PCR, so we can correlate with the phenotype of the EVs. The hope is that we will provide a toolkit that would be enabling for the community, and at the very least, very complementary to the methods that people would use. A method that provides single EV resolutions that can really delineate all the populations and subpopulations of EVs, be able to sort them, and then be able to subtype them using multiple markers like what flow cytometry provided for cells and immune cells. And then, so that's on the technology end, and then on the biology end, we hope that we can take one model EV, in our case, we hope to look at the semen exosomes, that we can completely provide all the subtype information there.